So I'm going to be talking today about utilizing iPlant to unearth long non-coding RNAs and then using CoGe to characterize their evolution in the plant family Brassicaceae. Okay, so one of the things that really I never really appreciated until I started studying long non-coding RNAs is how little we know about the transcripts that are produced by any given genome. And what I mean by this is if you really think about it, protein coding genes are essentially the most well-known, and they are essentially the 1%. They're the elite in the genome. And there's a reason for that. And of course, it's because protein coding RNAs are computationally easy to predict. And a large number of them have been identified empirically. They're often conserved across large evolutionary distances. I should mention that they're computationally easy to predict because they have markers for starting of translation, they have markers for stopping, they have poly-A tails, 5' caps, et cetera, et cetera. These protein coding genes can be functionally categorized based on the structural motifs that the proteins form. But as I said, protein coding genes really only constitute about a 1% or a very small percent of your genome. So what's going on with the other 99%? LincRNAs are not taking up genomic space. They're not just random aberrants of transcriptional noise, but a lot of these RNAs are, it's becoming more and more apparent that these RNAs are performing real biologically significant functions. Okay, so on to the transcriptomics. So again, this is the part where I really abused iPlant. I thought I was pretty proud. I was talking to somebody from TAC on Saturday night, and I was talking about how I had 2 terabytes of data, or maybe 3 terabytes of data in iPlant, and they're like, oh, that's nothing. But you know, it felt like something to me. But anyway, so basically, within iPlant, within the discovery environment, I assembled RNA-seq data, publicly available RNA-seq data from these 4 species. And here I'm showing you the number of loci that were identified in the genomes of these 4 species. Can we assume that conservation correlates with transcription? So what we did is we took a random sampling of 15 of those 92, and we asked, are they expressed? The ultimate answer is that yes, so I think, 12 out of the 15 transcripts, or loci, sorry, that we predict to be conserved lncRNAs are transcribed in Aethionema. So ultimately, we can say that if a lncRNA is transcribed in Arabidopsis and it's conserved throughout the family, then there's a really good chance that it's going to be expressed in the earliest diverging lineage. So here's the part where I get to iPlants, and some of you older people, like my age or a little bit older, will maybe recognize these screens. So my experience with command line, I kind of can echo Donovan's thoughts along these. My experience with command line before iPlant was playing these video games in MS-DOS. Most of you guys don't even know what MS-DOS is anymore, but these were like on 5-inch floppy drives, you know, that kind of stuff. So the beauty of iPlant and the discovery environment in particular is that you don't have to know any command line. I mean, as we've already seen, it's graphic user interface. It's really easy to run. If you do know the command line, if you are savvy with command line, then you can go into Atmosphere and take advantage of that. But for me, all this work that I showed you was performed in the discovery environment and in CoGe. And so, yeah, the biggest thing for me is, well, the second biggest thing, we'll get to the biggest in a second. The second biggest thing for me is the ease of use with minimal to no required skills. Getting ahead of myself, sorry. It's also easy to keep track of experiments and repeat problematic assays. And as we heard previously, RNA-seq is not a defined art. There's a lot of like hit or miss, trial and error, and so you repeat yourself a lot. And it's nice to be able to quickly go in, repeat an analysis, and slightly change some of the parameters. And so that's definitely a plus for me. Because of the computational resources, analyses take hours, not days or weeks. I actually did a kind of side-by-side comparison with an RNA-seq analysis on iPlant, and then I did it on the lab Mac. And I got bored after a couple hours because iPlant was already done, and the Mac was just still like 10% and kind of hanging. Analysis parameters can easily be shared. We've already heard about that. Pipelines can be created to kind of streamline your data analysis. And I think what is truly the most important thing, and I think that's why we're having a workshop here, and that's why these people have been so helpful and they're always there for you, is that this iPlant consortium is kind of founded or run by a bunch of extremely helpful scientists and programmers who are really only an email for me. And luckily for me at U of A, they're kind of, you know, across a busy intersection away. So I'll last leave you with a little tidbit or statement saying that really the information I've shown you today represents the tip of the iceberg in terms of plants' lincRNA characterization and evolution.